We have three talks in the first session. I know if you had coffee, but let's brew some more coffee with Debayan.
Okay, thank you so much. I hope I can make this coffee interesting. So, so today I will talk about how we went about brewing coffee, which is a general purpose force field for biomolecular simulations. So this is actually the work that was done when I was a postdoc in Dave Thrumalau's group at University of Texas at Austin. And another very talented postdoc, Balaka Mandal, was also involved in some of the work that I will show you. So the question that I pose is, somehow this is not working. Okay, so what really makes a nice cup of coffee? So that's a question, just to motivate. Of course, the quality of the ingredients, it matters, right? You need to really have very nice quality of coffee beans. You need to have good quality of sugar, milk, etc. And it's just not the quality of these individual ingredients themselves, but also a judicious combination. That makes for a nice cup of coffee. And of course, I'm not really trying to advertise Starbucks or CCD because that's not the purpose of the talk, as you would have guessed from the title. So for the purpose of my talk, here coffee basically is an acronym for this. Coarse grain phosphate for energy estimation. So that's what coffee means, for the purpose of this talk. And I will show you that even here, ingredients and their combination matter. Okay? So now you might ask, why was this force field called coffee? How we came about this name? So I think it's an important question, like many people were asking me during the discussion, what coffee means here. So to tell you a very simple answer, we wanted to keep the tradition alive. So if you look through the literature for the last couple of years or 5-6 years or so, it has been a practice of naming the different force fields that people have developed after popular drinks. So there is one called martini, one called absinthe, lassi, and so on. But luckily for us, no one used the name coffee. So we stuck to it. Okay. Okay. So the other question is, why did we choose a coarse graining world? Well, of course, there is a temptation always to use a very high resolution model to understand microscopic details when you are looking at a macromolecular complex. So this is a representative atomistic model of a certain protein, where as you see the protein molecule, the water, and the surroundings are represented in all atom detail. And so if I actually read, like, there's a lot of advancements in computer hardware that actually allow you to look at these kind of high resolution models of biomolecular complexes. But the problem is, when you're really interested in long time scales and large length scales or long time scales of biological interest, often this kind of approaches may not work because you do not really have that much of computer power. So an alternative route is actually to use what is called a coarse grain model. So what that does is there is a massive reduction in the number of degrees of freedom, as you can see from this picture. And what that allows you to do is actually to probe the system over much longer time scales and length scales. So what does this coarse graining do effectively in terms of the landscape? So what it does, it kind of smoothens out a very bumpy landscape that you would originally get for the all atom representation. So by that I mean it basically gets rid of many of this irrelevant minima. So it becomes a much smoother landscape as shown here. But the important part is any robust coarse graining method is it still should preserve the key features of the landscape. As you can see, for what I mean by that is some of these important minima that you see here should still be reflected in this kind of landscape, and once that is done, the underlying physics is still okay. So in the coffee that we are trying to brew, what are the key ingredients? First is a coarse grain model of DNA, then a coarse grain model of protein. So I will discuss what these are in the next few slides. And the idea is to sort of judiciously combine both these ingredients so that we can have a coffee or Starbucks or CCD grade. Okay, so that is the whole idea of this talk, how we went about combining these different ingredients and create this coffee model. Now, talking about the first ingredient of our model, this is the 3 interaction site model of DNA, where we represent the DNA in this kind of a coarse grain representation. So if you zoom in, here each nucleotide that you see is represented using 3 interaction sites. So one is centered on the phosphate group, Another one is centered on the sugar group as shown here, and the other one is on the nucleobase. So there are 3 interaction sites per nucleotide. And the potential energy function looks like this, where there is a term corresponding to the bonded interactions between the different moieties. There's an angular term like you see in phosphenes. There's an excluded volume term that ensures basically things don't come on top of each other. This one is electrostatics, which defines the interactions between charged phosphate groups. And the key interactions are basically these two, the stacking interactions and hydrogen bonding interactions, which is key for maintaining the stability of your DNA molecule. Now, the important point to note here is the stacking term and the hydrogen bonding term, as well as these two, both sequence dependent. Meaning that it will depend on what exactly nucleotide sequence you have. And this is based on this very fundamental work done by Santa Lucia and others in the context of lowest number model of DNA thermodynamics. So that is the starting point here. And the hydrogen bonding here, basically your AT and GC base pairing will have different energy scales. So this basically contains sequence information. And we recently showed that with this model, we can predict a lot of important properties of both single-stranded DNA and double-stranded DNA. So here I show some representative examples. So here, for example, if you look at 2 different single-stranded DNA molecules, so poly-DT and poly-DA, so basically we consider different lengths of this poly-DT and poly-DA, and the idea was to actually extract the effective Flory scaling exponent nu. And what we find is, if we look into how this exponent nu varies with cell concentration for poly-D and poly-DT, you see that the values are quite different, and it is consistent with what people have predicted from experiments, which are shown here is open symbols. And the important part here is, no matter which cell concentration that you go to, poly-DNA goes to this random coil limit, which is around 0.588. But for DT, actually, you sort of cross this limit at intermediate salt concentration. So our model kind of captures this sequence-dependent behavior of poly-DA and poly-DT sequences. And then we also looked at DNA hairpin melting thermodynamics for 4 different hairpins. So as you can see in this table, the last the lighting temperatures that we have predicted from simulations, we have a very good agreement with what others have reported from experiments. So basically it shows that this test DNA gives you a robust description of DNA thermodynamics and other properties. Next, coming to the second ingredient of our coffee model, which is the self-organized polymer model of proteins. So here each amino acid is represented using 2 sites. One which is, as you can see, which is centered in the C-alpha group, C-alpha atom of the amino acid residue, and one which is centered on the center of mass of the side chain. So this is basically a Coase-Gunn representation here. So here the potential energy function looks like this, where there's a Feeney potential which describes the chain connectivity between your different amino acid residues. There's a repulsion term which again prevents unphysical chain crossing, and these are the 3 different very important interaction terms which defines the non-covalent interactions between the side chains, C-alpha, C-alpha, and C-alpha side chain beads. And this is of course the electrostatics part, which describes the interaction between charged amino acids. And these 3 potentials again are basically of this Lennard-Jones form, and this epsilon has different values depending on whether you are considering a side chain, C-alpha, or C-alpha SC interactions. Now the important point to note here is this side chain interaction. So this is the part of this SOAP model that actually encodes the sequence specificity. So this is how it is in fact written. And this epsilon I that is shown here is basically called the Betancourt-Through-Malan matrix. And this epsilon I will take different values for different amino acid pairs that are in contact. So this is where the sequence specificity part of the model comes in. And this is actually a very well-established model with Dave's group and others. They have used extensively to actually look at protein folding problems. For example, here what I show is the folding of ubiquitin. They were able to show that it follows different folding pathways based on the pH conditions. And here, Reddy and others, they were able to look at the folding pathways of green cells and proteins. So this model was mostly developed by Dave and others. Coming to coffee, how do we combine these 2 different ingredients? So one can write down the potential energy function for the protein DNA interaction in this way. You have the test DNA model, the SOAP-SC model, which I would explain in the last 2 slides. And then you also have additional interactions term which describes the interaction between your DNA and protein molecules. Now one can decompose this DNA-protein interaction into electrostatic and non-electrostatic components. So electrostatic is basically where your charged amino acids kind of interact with your phosphate backbone. And then there's a non-electrostatic component which one can write down as coming from native-like interactions and non-native interactions. So here native interactions, I mean, where two given residues are already in contact in a reference structure, which could be an X-ray structure or an NMR structure. So typically this is like a Mo interaction, just to put it simply. And this laminar DNA approval is basically sort of controls the strength of this DNA protein contact potential. And residues which are not in contact in the reference structure, we consider them to be non-native, and these interactions are purely repulsive. So this is how the native interaction is represented. So as you can see, this is basically the reference distance in the crystal structure at the NMR structure, as the case may be. And the important part here is this energy scale epsilon xy. And this is again different depending on which two residues you are considering. And the way we derive these energy scales were from PDB contact statistics. So what we basically did is we looked at a database of longitudinal data set of DNA protein complexes, which was published by these guys. And we basically, it was just then after that a counting exercise. We counted the number of times each of the different residues of forming contact. So and we chose a certain cutoff distance to define a contact. And once you do that, basically the energy scale then becomes typically the free energy cost of forming a contact between residues X and Y, which you can write in this way. So PLGXY is the probability of contact formation between beads X and Y. So this X could be either a phosphate, a sugar, or a VPO-based bead, and Y could be either a C-alpha or a side chain. And IJ is basically the type of amino acid and the nucleotide involved. So I could be any of ATGC, and J could be any of the 20 amino acids. And here the thing is, this is often called the statistical potential. So the important part here is the choice in the reference state. What you choose as the reference. So for simplicity, we assume that the reference state is nothing but the random chance that a given pair is in contact. So in that case, it will simply be the product of the individual probabilities. And these kind of approaches have actually been pioneered by several groups, most notably by Harold Shiraba's group. So they were the first one to develop these kind of statistical potentials to study protein folding right around 1975. Okay, so next, as you can see, there is one tunable parameter that we have in the DNA protein interaction term, because we are not touching the DNA potential or the SOAP-HC potential. We are keeping everything fixed. So the only adjustable parameter in our model is basically the lambda DNA probe that I talked about, which tunes the strength of your DNA protein contact potential. So how did we optimize it? So what we did was we considered a very high resolution DNA protein complex, which is this human TVP code domain complex with DNA. So it's a very small structure, as you can see. And we tried to vary the strength of this lambda DNA probe. And then we computed the root mean square error between the P factors that are reported in the X-ray structure and the B factors we estimated from simulations. And as you can see, like if we plot the root mean square error for different values of this lambda DNA probe, the optimal value turns about around 0.5, and beyond that we do not see any really any change in the observations. And this is just to show how for the optimal value of this lambda DNA probe the comparison looks like. So you see that many of the values look okay, but there are some large deviations near the termini, which because of the enhanced fluctuations that you could see in your simulations. And the other fact is, of course, in crystal structures you have this crystal forces which we do not have in our simulations. So how does this coffee actually perform when we consider different DNA protein complexes of different sizes? So we actually considered 3 different complexes of different sizes. Here I show you a comparison of the simulated on the experimental SAC spectra. So the experimental SAC spectra is shown here as gray circles, and the one that we estimate from simulation is shown as this red curve here. And as you can see, these are all 3 different lengths. And the important part here is, as you can see, that the radius of gyration that we estimate from simulations and the one that is estimated from SAC spectra are pretty much in quantitative agreement, and even the SACs sort of agrees pretty well to reasonable ranges of Q. Now, this is all about the global dimensions of DNA protein complexes. I mean, that is what SATS tells you mostly. But how about the local structure? How is coffee doing in terms of the local structure? So for that, we actually looked into this one operator DNA complex with the lab pressure, and we tried to compare those chemical shifts that we compute from our simulations with experiments for these two different domains of the DBD. As you can see, again, the chemical shifts are pretty much very close to what you get from experiments, and this is where I show you the contact maps for the NMR ensemble and from the simulated ensemble. As again, you see that many of the signatures that you see in the NMR ensemble are actually retained. Our actual motivation of trying to develop this model was to understand some aspects of genome organization. So let me just give a brief overview of what I mean by that. So as you know, we have about 2 meters of DNA, which is compressed into a very small volume of the nucleus. And how it happens is basically through different layers of hierarchical organization. So the first layer of organization is the formation of this biomolecular complex, which are called nucleosomes. So in nucleosomes, you basically have about 147 base pairs of DNA, which is wrapped around an octametric core of histone proteins. And then these nucleosomes can sort of form an array, which is called a chromatin, which can have further folding mediated by different proteins, and ultimately you get to this chromosome. So our idea was to just understand the structure and the dynamics of this nucleosome core particle. So unlike what is shown here in this figure, nucleosomes are actually quite dynamic. So they can basically do this. Some of the DNA can actually come off, and this is what is called the wrapping transition. And there are several groups, most notably John Widdom's group, they have studied this to great detail, and they have shown that it has important consequences when it comes to transcription. And very recently, there was a very interesting work done by Louis Polak and co-workers, where they showed using experiments that if you keep on increasing the cell concentration, you can actually completely disassemble the nucleosome. For example, when you are at very low cell concentration, the nucleosome is basically intact, but then once the cell concentration increases, you induce some bleeding motions in the DNA, and then there is a loss of different numbers of histone proteins, and finally at around 2 lower cell concentration, there is a complete disassembly of the structure. Another very interesting study in this direction of nucleosome unwrapping was from T.J. Howe's group using single molecule peeling experiments combined with FRET. So what they showed is basically the DNA nucleosome wraps in an asymmetric fashion. In other words, if it depends on the local DNA sequence around these wraps. So if your DNA sequence is somewhat more flexible, it will basically be accommodating this bend, and you require a much higher force to pull it out. But if your DNA end is less flexible, depending on the local DNA sequence, it will come off easily at a much lower force. So in other words, there is some kind of asymmetry that is there. Similarly, following up this work, there was again some very interesting studies from Polak and co-workers where they tried to induce this unwrapping not by force but by just changing the salt concentration. And here what they observed was like there are different possibilities. You could have a pathway through this symmetric state, and then there could be also this pathways through asymmetric unwrapping, which was observed by T.J. Howe's group. And interestingly, around 2018, a couple of years ago, Halleck and co-workers published some cryo-EM structures of various stages of nucleosome unwrapping. And as you can see, in all these structures, the unwrapping is asymmetric. So we wanted to understand actually the finer details of this nucleosome dynamics, primarily this unwrapping process. What is the origin of this asymmetry? And is it like a universal phenomena irrespective of the DNA sequence that you have? So that was one of the goals. So the first step in this direction was we wanted to see if our model is actually giving a good enough description of the nucleosome structure itself. So what we did was we computed the SAT spectra from our simulations, which is the one shown in the red curve as the red curve. And these green points are from experiments. And here, as you see, these are the reported values from simulation and experiment, and they're in very good agreement. And even the SATs, as you can see, it sort of is pretty much on top of the experimental SATs profile. So far, so good. And then we wanted to see what happens if you increase the salt concentration. Are we able to see something interesting? So here these are basically the probability distributions in terms of the number of unwrapped base pairs. So I'm not going into the details of how we actually calculated the number of unwrapped base pairs because it's a bit involved, but I'm happy to discuss it after the talk. So as you see, like when you are in a very low salt concentration, around 100 millimolar of sodium chloride or so, your nucleosome tends to be fully wrapped. So there is no unwrapping what is happening. But as you keep on increasing the salt concentration, you see more and more metastable states have exhibiting different degrees of unwrapping being populated. And interestingly, what we see both partially unwrapped states which have this symmetric unwrapping, meaning that there's the same extent of DNA coming off from both ends. And we also see some asymmetric unwrapping where it's just one of the ends which is sort of coming out. And interestingly, this population on this asymmetrically unwrapped states is somewhat more than what we see for the symmetry. Interestingly, what happens is, maybe I did not explain it well, if you say in the nucleosome structure there are this kind of tails. So these are called histone tails, and these are intrinsically disordered in nature, and what they do is they mediate internucleosomal interactions when you go to the chromatin array. So we wanted to understand what happens if we delete these tails. Does it actually impact the unwrapping process or the mechanism? So what we find is when you delete these tails, no matter what cell concentration you are at, even at very low cell concentration, your nucleosome is not stable. So there is a population of partially unwrapped states. And similar to the previous example, when we had the tails, we see all these different kinds of unwrapped states showing both asymmetric and symmetric unwrapping. And interestingly, when you are at around 1 molar, there is a full disassembly. So this entire histone core actually comes out, which we don't see in the previous case when the tails are there. Now, here I show you the average number of base pairs that are unwrapped for the case with and without tails. So as you can see, with tails, effectively, when you are at low salt, the degree of unwrapping is very low. But when there are no tails, there is always some degree of unwrapping that goes on. Okay, so now the next question was asymmetry. Can we talk about asymmetry from our model, and do we see any degree of asymmetry? So to measure the asymmetry, we define this parameter gamma, which is nothing but defined in this way, where this is basically the number of base pairs that are unwrapping from the left direction. So you can basically define it in an arbitrary goal, which one is left, which one is right. And then this is the total number of base pairs that have unwrapped, and this defines the asymmetry. And then we looked at different cell concentrations. So when we are at somewhat low cell concentrations, around 200 millimolar, you see there is an equal chance of having the symmetric and the asymmetric states, as you see from this free energy diagram. However, when we push the salt concentration to 500 millimolar, one of the states or one of the sides start to win. So as you see, there is some permanent asymmetry where there's a higher chance of actually unwrapping it from the right as opposed to the left. Interestingly, when we remove the histone tails, when we chop off the histone tails, we find that the asymmetry becomes much, much weaker, even at moderate salt concentration. So the reason being is this, like when a substantial amount of the DNA has actually detached from your nucleosome core, there is some communication that is sent to the other end, and the histone tails kind of hold the other end and prevent it from unwrapping. Okay, but when you chop up the histone tails, those interactions are lost. So effectively, the asymmetry is lost at moderate salt concentrations. So the histone tails basically act almost like a gatekeeper. So we are still kind of in the process of understanding this asymmetric process in detail. There are certain things which we do not really understand at the moment. Now, the other question we asked is whether we can prove the effect of mutations on nucleosome flexibility. So for that, what we did is we mutated some of the arginines that is shown here into lysines. And the reason being, these arginine phosphate salt bridges, they are going to be a very key component of nucleosome stability. And disruption of these contacts are actually associated with certain mutations in yeast, and they are known to sort of enhance the nucleosome flexibility. So we wanted to see if our model is able to capture this or not. So this is a distribution again in terms of the unwrapped base pairs I show for the canonical nucleosome, where all these contacts are arginine phosphate. And for the mutated nucleosome, where these arginines have been sort of mutated into lysines. So as you can see, for the canonical nucleosome at 500 millimolar, there is a bimodal distribution, where the major peak is around 20, but when we mutate it, it's still sort of bimodal, but now the population shifts and the major peak is around 70. So it is effectively making the nucleosome more flexible. The other important aspect here is, as you can see, this is largely a chemical effect, because lysines and arginines, they basically have the same charge. So many of these existing close-grained models that are there mostly will not be able to capture this, because this use the same kind of energy scale for lysine and arginine contacts. So this is one key or unique feature of our model. I mean, this is only coming about because we are using this sequence-specific interactions for the DNA and the protein contacts. Now, what are actually we doing when we are not brewing coffee? So there are different things which we are thinking about. So one of them being developing minimalist models for intrinsically disordered proteins. And the idea is to probe their energy landscapes to understand their conformational heterogeneity and so on. So this is the work which we are doing in collaboration with Pinaki. And then the other problems we are thinking about includes playing around with elastic network models to probe the role of allosterine in the case of substrate binding of cytochrome P450. So this is the work currently being done by Tahir, who recently joined as a postdoc in IMSC. And then we are also trying to probe the mechanisms of RNA protein binding, and this is what we are doing with Vani. So just to sort of summarize, we brewed coffee basically by combining two robust coarse-grained potentials for DNA and proteins in a judicious manner. And I also showed that we are able to recapitulate the SAC spectra reported from experiments and also some aspects of salt-induced destabilization of nucleosomes. Interestingly, what we find is the salt concentration seems to modulate the direction of unwrapping as well as the degree of asymmetry, but this is something which we're still not completely in grips with. And we also showed that our model, because it has this sequence-specific interactions between the DNA and proteins, it captures the effect of mutations on nucleosome stability. And so, just to conclude, I would give convey a big thank you to Chennai Soft Matter Board organizers, Pinaki and Arnab. And I should be very happy to answer any questions. And I hope I have finished on time.
You calculated the melting points, and the higher the CG content, the better the accuracy was.
Yeah.
So why is it so?
Okay, so that is a good point, but I think it's just a matter of coincidence here. I don't think there is anything in the model that will tell you why there's a higher GC content. It should be agreeing better with experiments. So the idea is, of course, the melting temperature will depend on the GC content. It tends to be higher. There is all these Marlowe-Doty relations and so on that people know of. But I would say here it's just a matter of coincidence that why for higher GC content we are getting a better agreement. And of course, we have just looked at 4 sequences. I mean, if we include more, and we haven't really done a systematic study of, sort of scanning through this entire range of GC content and then doing the comparison with our model. So before that, I wouldn't comment on it, like why. I think it's just purely coincidental in this case.
Yeah. So, nice talk. And I couple of questions. One is on the effect of ionic strength or the salt concentration.
What?
How do you include that in your...
Okay, so that is just using simple Debayan actually.
Right. It's not generally applicable beyond about...
Yes, that is a good point. So that is why we have a model where we are actually including explicit ions to model those effects. So it's a good point. It won't be
So you have included explicit ions in the...
Not in the results that I have shown you here.
Okay.
Yeah,
Okay.
yeah.
And the second... So you do not include explicit ions in the model, but how are you then reproducing it? Because you tweak the devehicle such that it can capture some effect of the salt.
Yes, we tweak the screening length actually.
Which one?
The Debayan screening length.
Okay.
Yeah.
Okay. The screening length you modified.
Yeah, yes.
That was modified based on...
The salt concentration.
No. I mean, what experimental...
Yeah, exactly. So we've actually looked at the experimental salt concentration, the monovalent salt concentration. That is how we tuned it. That is what we set it to actually.
Okay.
Yeah.
So the other point is on the arginine lysine mutation. So, and the fact that the mutation captures it, I would be surprised because in your model, this is not atomistic in some ways. So it's only the contact potential that is varying. Really, right? So in your probably a thermally Bentacote model, it is the contact potential between lysine and the other residues that are varying compared to arginine versus other residues. Is it not? Because how is it otherwise capturing
So
the...
so this F7 XY that I showed here, let me just go back to the slide. So this one you see where this F7 XY energy scale. This is actually different for lysine phosphate contacts and arginine phosphate contacts. And this is basically from when we looked at a database of high resolution structures, we found that is how it is. Like, there are more, the arginine contacts are more favorable with nucleobases or the DNA as compared to the lysine contacts.
So that is reflected in the epsilon XY.
Yes, exactly.
It means that epsilon XY is more negative for the arginine.
Exactly. Yeah, it's more favorable. The free energy is more favorable.
See, one final question. So it's on the solvation. You do not consider solvation explicitly, but then dielectric constant is temperature dependent.
Yes, dielectric
You consider a temperature dependent.
Yes, we do. We do consider the dielectric constant to be temperature dependent.
There is also this, of course, the solvation term which is not included.
It is not included. So we are actually thinking of having a better description of the solvation. So what we are trying to do is now introducing an implicit solvent model based on the generalized bond formalism. So we want to, because in many aspects when you see this, desolvation and dewetting, so those kind of things we won't be able to capture. But hopefully when we have some description of the solvent model, better description in terms of generalized bond or anything equivalent, maybe we have a better handle of it. And the other approach we are trying to do is trying to include some coarse-grained model of water. Like for example, this water models that Valeria Molinares group, they have developed. So we are trying to integrate those models with this framework.
Continuing on this question, does that epsilon XY capture the protonation states and the difference that comes because of pH changes and things like that?
No, it doesn't.
Okay. Which is there any term that captures?
Not yet. So what we can do is this. So if you want to do it on a different pH, you can change some of the protonation states of the amino acids without touching this part. So that will be taken care in the electrostatic.
Protein.
But what you can do is, even like you remember, there's an electrostatic component to the DNA protein interaction as well. So based on the pH, you can assign the appropriate charges to our amino acid side chains. That would be one way of doing it.
Yes.
But the other thing which we are doing is, effectively, let's say we do a simulation at pH 7. How do I know what's the conformational ensemble at pH 2, for example? So there are some reweighting techniques that we are developing. So reweight the simulations at different pHs and try to understand how the landscape evolves and so on. So yeah.